Good morning, everyone. We're back once again with another amazing science tutorial video. I'm Coach Spivey, joined with my son Jordan Spivey, and in this video, we're going to go over some of the biology concepts that we've covered so far. So let's begin. So let's go over one of the first concepts, and that's Mendels Law of segregation. And Mendels Law of segregation states that chromosome pairs separate during meiosis so that each gamete receives one allele for a given trait. So for example, we start off with these chromosome pairs right here, but if you notice, as we go through meiosis 1, they begin to separate. So they separate this pair right here and then into this pair right here. So I go ahead and highlight that. So they begin to separate. It says during meiosis so that each gamete receives one allele for a given trait. So if you notice, as we continue through meiosis 2, it shows that each gamete has received one allele for that given trait. And now let's take a look at Mendels Law of independent assortment, which states that genes for one trait are not inherited together with another trait, which means that most genes don't come linked together. So for example, if you look at this heterozygous cross between two traits, we're looking at P color and P shape. Notice that the P color and the P shape don't come directly linked with each other. So some of these P's are round and yellow. Some of these P's are round and green. And then if you notice, some of these P's are wrinkled in yellow, and then some of these P's are wrinkled in green. And this is a perfect example of showing that traits are not inherited together. They independently assort from one another. So now it's time for your check for understanding. Answer the following questions on Mendels laws and pause the video to answer each question. Now let's check and see how you did. In our first problem, it says only purple roses come from a cross between homozygous purple and homozygous white roses. This demonstrates the principle of, if you notice, we have purple flower right here crossed with a white flower, and our F1 generation, our first generation, is all purple flowers. So let's go ahead and cross out what we know is not. So we know it can't be independent assortment because we're only looking at one trait. And then incomplete dominance would actually show that there's a combination of both traits. So it would show that it may be pink. So the only answer that it could be is dominance, because if you notice, the purple flowers are dominant over those white flowers. Let's look at our next one. Which of the following is true about the crossing over process during meiosis? So A, it occurs between sister chromatids. No, it can't possibly be occurring between sister chromatids. B, it prevents genetic recombination. Well, actually, if you see right here, when it's crossing over, you're looking at that genetic recombination right here. So it can't be B. C, it happens only once per homologous pair. No, it can't be that one. So our most likely answer is going to be D. It increases variability in gametes. And the way it does is that when it crosses over, when these non-sister chromatids cross over, that increases the amount of genetic variety or genetic variation. And then on our last problem, a cross between two rabbits with long tails and gray fur produced the four types of offspring as listed: long tails and gray fur, long tails and brown fur, short tails and gray fur, short tails and brown fur. This best demonstrates which of the following genetic mechanisms. Well, if you notice, in this example, we're looking at two traits. We're looking at tail length, and we're also looking at fur color. And if you notice, in the offspring, none of them come together. None of the traits actually come together. And the way we can tell is because we have some with long tails and gray fur, some with long tails and brown fur. We have some with short tails and gray fur, and short tails and brown fur. So this best demonstrates the law of independent assortment. And if you look at it, if we go back to our example of peas when we was looking at yellow or green and round or wrinkled, notice that they independently assorted, and so yellow didn't always come in round and green didn't always come with smooth or wrinkled. So that's how we can tell with this one, because long tails doesn't always come with gray fur and short tails doesn't always come with brown fur. They independently assort. So now let's look at some Punnett squares terminology. And we're going to be looking at comparing complete with incomplete dominance. And if you look at complete dominance, the dominant allele completely masks the recessive allele. So here's our homozygous dominant red flower crossed with a homozygous recessive white flower. And if you notice, that homozygous red or homozygous dominant red flower completely masks that homozygous recessive white flower. So that's why all of the offspring are red. And then if we look at incomplete dominance, neither allele is dominant. So here's that homozygous dominant red, but then here's the homozygous recessive. But if you notice in the offspring, neither one is dominant, because we can tell all of them are not red and all of them are not white. More so, they are actually a cross between each other, and that's why we end up with these pink flowers right here. So what is codominance? Codominance is a condition in which both alleles of a gene are expressed in heterozygous offspring. So for example, in certain chickens, black feathers are codominant with white feathers. And what this means is that you're going to see both phenotypes. You're going to see the black feathers and you're going to see the white feathers of the chicken. So heterozygous chickens have black and white speckled feathers, just like these chickens over here to the right. Let's take a look at some Punnett square practice problems. So if a heterozygous purple flower was crossed with another heterozygous purple flower, purple is dominant with white being recessive, how many of the flowers would be purple and how many of the flowers would be white? So let's go ahead and set up our Punnett square. So we said they are heterozygous for purple. So let's underline that because that's important information. So that lets me know I'll put a capital P, lowercase p, capital P, and lowercase p. And this shows that heterozygous cross. So let's fill out our Punnett square. We have a homozygous dominant here. We have a heterozygous dominant right here. Heterozygous dominant here, and then we have a homozygous recessive right here. And it asks us how many of the flowers would be purple. Well, we notice if there's at least one capital P, which is that dominant trait, then the flower is going to be purple. So let's go ahead and count the ones that have at least one capital P. So this one right here will be purple, and I'll go ahead and change my pointer option, just change it to purple to let us know what color it'll be. So this will be purple, this will be purple, and then this would be purple. So to answer that first question, how many of the flowers would be purple? So I'll do this, purple equals 3. Then our next question, how many of the flowers would be white? So in order for the flowers to be white, since white is recessive, you would have to have 2 lowercase p's. And so let's go ahead and count the number of ones that have 2 lowercase p's, which would be this one right here. And so I put this mark right here. I know it's hard to see, but I also put it over here. So how many of those would be white? There would only be one that would be white, a white flower. And then in the Punnett square for flower color, what is the probability that an offspring's color will be white? So let's go ahead and fill out our Punnett square again. Let me go ahead and change my point option so you can see the ink clearly. So we had a homozygous dominant here, we had a heterozygous dominant here, we had a heterozygous dominant here, and then we had a homozygous recessive right here. And it asks, what is the probability that an offspring's color will be white? Well, there would be a 1 out of 4 chance, because 1 out of the 4 flowers are white. And then on to our last problem. In the Punnett square for flower color, how many of the squares show the genotype of homozygous dominant, heterozygous, and homozygous recessive? So let's go ahead and fill out our Punnett square again. So here's that homozygous dominant, here's that heterozygous dominant, here's another heterozygous dominant, and then here's that homozygous recessive. And so how many of them are homozygous dominant? That means they have 2 capital P's. Well, we have one that has 2 capital P's, so here's 1. And then how many are heterozygous dominant? That means that we have a capital P and a lowercase p. We have 2 with a capital P and a lowercase p, so I'll circle it right here. And then I'll go ahead and circle it right here. So there's that 1, 2. And then how many are homozygous recessive? We have 1 that is homozygous recessive. So our ratio, it would be 1 to 1. So our answer would be B. And now let's take a look at the advantages and disadvantages of sexual and asexual reproduction. So one of the major advantages of sexual reproduction is that it increases genetic variation. So offspring can adapt and evolve, and it increases their chances of survival. So this means you have many different combinations of variations of genes and traits, which increases that offspring's chance of survival. A disadvantage of sexual reproduction would be that it's slower than asexual and more stages involved. So that means it takes time for sexual reproduction to occur. And then energy and time is used up to compete for a mate and also for the sexual reproduction part. Now let's look at asexual reproduction. So benefits of asexual reproduction would be offspring are genetically identical, and this is good when the parent is well adapted. And then since they're genetically identical, it's quick. So bacteria can produce offspring in 20 minutes. So since they're genetically identical, they can just make exact copies of themselves, and it's very quick, and they don't have to use a lot of energy and time. A disadvantage of asexual reproduction is all faulty genetic material is passed on. So whatever faulty or whatever is wrong with the material before in the first generation will get passed on to the next generation. Another thing is that one thing could wipe out that whole generation of bacteria since they're all genetically identical. Now let's look at some of the differences between viruses and cells. And these are some of the main differences between cells and viruses, and they're summarized in this chart below. So if you look at the structure of a virus, it has DNA or RNA inside of a capsid and some that have an envelope. And then if you look at the structure of a cell, the cell membrane, their cell membrane cytoplasm, eukaryotes also contain a nucleus and many other organelles. And then if we look at the reproduction of a virus, they can only reproduce within a host cell. Reproduction for cells, the independent cell division, either asexually or sexually, they don't need a host cell in order to reproduce. They can reproduce on their own. And then the genetic code can be DNA or RNA for viruses. The genetic code for cells is always DNA. Viruses do not grow and develop, and cells do, especially in multicellular organisms. So cells increase in number and they differentiate. That means you have different cells for different functions, such as your liver, your bones, your heart cells. So all the cells are going to be different. And then viruses don't obtain and use energy. Cells do have to obtain and use energy in order to survive. And then viruses don't respond to the environment, while cells do respond to the environment. So if there is too much solute on the outside of the cell, the water rushes out. If there's too much solute on the inside of the cell, the water will rush in to balance and respond to that environment. And then viruses do adapt and change over time, and so do cells as well. And now let's do a quick review over phylogenetic trees. So number one, what type of organisms do all of the other organisms originate from? Well, if you look down here, and I'll change my pointer option. If you look down here, all of these organisms originated from prokaryotes. And then number two, what are worms closest related to? So let's go ahead and identify or locate where worms are. So worms are right here. And if you notice, they come off this same node or this same branch as the mollusk. So they're closest related to mollusks. And then number three, which are more closely related? Chordates and mollusks, or chordates and hemichordata? So let's go ahead and locate our chordates. Here's our chordates right here, and here's our mollusks. And then let's go ahead and locate our hemichordata. And if you notice, even though the chordates and mollusks may appear to be closer right here, look right here, the chordates and mollusks branch off on different branches. So the mollusks are over here, the chordates are over here. And if you notice, the hemichordata are actually on the same branch as the chordates. So which are more closely related? That would be the chordates and the hemichordata. Then number 4, which organisms are number 4, which organisms are Cnidaria least related to on their branch? So here are Cnidaria right here, and it's hard for me to say. But if you notice, they're the closest related to comb jellies, but if we go all the way across, they're the least related to Placozoa. How can we tell? Because they're the furthest apart from each other on that branch. And now let's look at our last concept, which is cladograms. So number 1, what trait separates lampreys from tuna on this cladogram? So let's go ahead and locate the lamprey. So here's our lamprey right here. Then here's our tuna. If you notice, both of them have a vertebral column, but what you notice is that the tuna actually has jaws and the lamprey does not. And number 2, what separates a salamander from a turtle? So if we look, here's our salamander right here, and then here's our turtle. Both of them have a vertebral column, jaws, 4 walking legs, but a turtle has an amniotic egg. So that's what separates that salamander from that turtle. And then number 3, which organism is most related to the leopard? So if we take a look, the leopard and the turtle both have vertebral column, jaws, 4 walking legs, amniotic egg. The difference between them is that a leopard has hair and a turtle does not. But they have 4 other traits in common. So the turtle is most related to the leopard in this cladogram. And then number 4, what 4 traits do these 2 organisms share? They both share a vertebral column, and I just put vert-col, just abbreviated. They both share jaws. They both have 4 walking legs. And then they both have amniotic eggs. And then with number 5, which organism will have DNA most similar to the turtle? So if we look, the turtle has vertebral column, jaws, 4 walking legs, and amniotic egg, but it does not have hair, so it won't be as similar to the leopard, but it would actually be most similar to the salamander, because both of them have their vertebral column, jaws, 4 walking legs. The biggest difference between the 2 is the amniotic egg. So it would be most similar to the salamander. And then number 6, which organism's DNA will differ the most from the leopard? If we notice, and I'll erase this so you can see, if we notice, the leopard is on this side of the cladogram and it has all of these different traits. And the lancelot has none of these traits. It's on the opposite side of the cladogram. So since the leopard has all these traits and the lancelot shares none of these traits, the lancelot will differ the most from the leopard. Ladies and gentlemen, I'm Coach Spivey, signing off with my son Jordan Spivey, and I hope this biology review was beneficial and helpful. Peace, and have a wonderful, awesome, positive day.